Let me to dictate you, because this is important. You will get the question. Definitely, you may get question. There are fair enough chances that you will get question from. Describe what are the photosystem, or what is the photosystem first, photosystem second, right? So put the heading photosystem, right? Photosystem or pigment system. That's the heading. Photosystem or pigment system. Start right now. In green plant, photosynthetic unit, photosynthetic unit occur in photosynthetic unit occur in form of two distinct groups called photosystem or pigment system. Photosystem or pigment system, first and second. Next line is very important. They are named after the sequence. They are named after the sequence they were discovered. They are named after the sequence they were discovered. Full stop. Each photosystem contain 250 to 400 pigment molecule. Now the first subheading will be PS1 or photosystem 1. PS1 or photosystem 1. They are located on first point. Put it write it down in the bullet form, right? Bullet form or in point form, not as a paragraph. They are located on non-appraised part that I have discussed already discussed in last class, right? They are located on non-appraised part of grana. Full stop. PS1 has more chlorophyll A molecule. This is the question for me. PS1 have more chlorophyll A molecule. Full stop. Chlorophyll B in the same line. Chlorophyll B and carotenoid. Chlorophyll B and carotenoid are comparatively in less amount. Chlorophyll B and carotenoid are comparatively in less amount. Next point. PS1 have photocenter. PS1 have photocenter and electron carrier. Photocenter is made up of two chlorophyll A molecule. Photocenter is made up of two chlorophyll A molecule called P700 called P700. Next, it also contains chlorophyll B and carotenoid. It takes part in both cyclic and non-cyclic. It takes part in both cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. Right? So this was the PS1. Clear?
Yes, sir.
Now come to the PS2. Right. So as I have told you, the diagram for both of them is same. Right. But there is a bit difference. What is the difference? We have to start from here. First, I will explain it for you. The diagram will be, as I have told you, that diagram will be same for PS1 and PS2. Photo system, they have same diagram, but there is only difference in the composition. So basically, that was present in PS1 was present in non-appressed part, means those part which are the outer region, right? Open region, right? Free part. Appressed part. So I have shown over here appressed part. So suppose, not suppose that this is basically thylakoid. This is, these are the basically thylakoid, right? Thylakoids. In thylakoids, those parts which are in contact with each other, right? The surface touches each other, right? There is a common surface. There is the surface touches each other. So these parts are called basically now appressed part, right? And the PS2 is found basically appressed part. And we have already discussed in detail in last class that in here the PS2 is found. And PS2 is responsible for the breakdown of water molecule. So the breakdown of water molecule is called photolysis of water or H2O. Photo means light and lysis means breakdown. So in the presence of light, breakdown of water molecule takes place. Oxygen and hydrogen ion come out. Along with that, electron also come out, right? This process is called photolysis. So basically, PS2 is responsible for photosynthesis. So how does PS2 break down the water molecule? To break down water molecule, PS2 have a special kind of the thing, right? Photocenter and electron carrier, that is a present in PS1 as well. But PS2 have one more important that is called OEC. What is OEC? Oxygen evolving complex, right? Oxygen evolving complex, right? So OEC stand for oxygen evolving complex. So basically, this OEC or oxygen evolving complex is made up of, it contain manganese, calcium ion, and chloride ion. This is the composition. That's how they are made up, right? And this OEC, oxygen evolving complex, this is responsible for breakdown of the water molecule, right? So here the chlorophyll A and B and carotenoid is found, right? And chlorophyll A, B both are present in equal amount. Here carotenoid is higher compared to the PS1, right? And photocenter is made up of P680. So that is the composition of PS2, right? And the what is the exclusive over here? That is the OEC or oxygen evolving complex or breakdown of water. Tell me, is it clear or not?
Yes, sir. It's clear.
Okay, so please first draw this diagram. Draw this diagram, right? And then I will dictate for you. Martin, are you getting everything? Done? Note down? You have not to note down this diagram. Don't note down this green diagram. I'm saying just note down this diagram. Alia, done?
Done, sir.
Yes, sir.
Okay, fine. So start noting down. PS second is located in appraised part of PS second is located in appraised part of halakoid. Appraised part of halakoid. Next one. PS second have chlorophyll A, B, chlorophyll A, B, and carotenoids. Full stop. Chlorophyll A and B are present in chlorophyll A and B are present in equal amount. Chlorophyll A and B are present in equal amount. Full stop. Next point. Carotenoid content is higher in PS2 as compared to PS1. Next, PS second contain photocenter, comma, oxygen evolving complex, and light harvesting complex. And electron carriers. Next point. Photocenter is made up of dimer of photocenter is made up of dimer of chlorophyll A molecule. Photocenter is made up of dimer means two. Dimer of chlorophyll A molecule called P680. Next point. Oxygen evolving complex OEC in bracket write down OEC. Oxygen evolving complex in bracket OEC contain manganese, calcium, and chloride ion. This is question for D. OEC contain manganese, calcium, and chloride ion. OEC is responsible for OEC is responsible for breakdown of water, and release of oxygen and electron. Release of oxygen and electron. Is that clear?
Yes, sir.
So I will tell you in NEET class that there are some photosynthesis where oxygen do not come out as a product. They are called anoxygenic photosynthesis, right? We'll discuss in NEET class. Fine. Now electron transport chain. What is the EPC or electron transport? Right. So look. So basically what happened, PS1 and PS2 both are responsible for carrying the electron and production of energy, right? So, okay, dark and light reaction that I have already explained. Look, there are two kind of electron transport chains, cyclic and non-cyclic. What is cyclic and what is non-cyclic? In cyclic, electron come back its original position. In non-cyclic, electron do not come back. Right? Give me 1 second. Look, first electron transport chain is that is the non-cyclic electron transport. And this is called photophosphorylation. So what is basically photophosphorylation? Photophosphorylation term denotes that formation of AT, right? So the nutshell, photophosphorylation means formation of AT, means production of energy. Okay, so this is the PS second. So first, what happened in PS second, photosystem second? The light falls on antenna pigment. These antenna pigment capture the light and give it to the photocenter or reaction center, right? Photocenter or reaction center, PS2. So first, the story start from PS second. Why? When the reaction start from here, why we call it PS second? Because it has been discovered later. The name of PS1 and PS2 have been given on the basis of their discovery, on the basis of sequence of their discovery, right? So it was first PS1 discovered and then PS second discovered, but the reaction it comes first, right? So this light energy fall over here, right? The light energy fall over here, it captures the energy and transfer to this core molecule called reaction center. It have the oxygen evolving complex OEs that we have already discussed. So when water molecule comes over here, water molecule break down, two hydrogen ion comes out, two electron comes out, and half oxygen molecule comes out, right? So finally, when two water molecule break down, then only one oxygen molecule comes out, right? Because half water molecule is half oxygen molecule is nothing, right? There is nothing half like, uh, but here numerically we call it half oxygen molecule come out, right? Then what happened along with the photolysis of water, when water breaks down, electrons also come. So electrons are received by electron acceptor. Then electron reaches to pheophytin. Pheophytin, they are the pigment molecule which are found. They are the basically protein molecules which are found in the membrane. Then it comes to the PQ, uh, pheophytin, right? Then it comes to the plastoquinine, right? Electron is transferred plastoquinine, we refer it for PQ. From plastoquinine, it comes to the cytochrome B6F complex, right? When electron come to the cytochrome B6F complex, formation of ATP takes place, right? Then what happens? This is transferred to the plastocyanin, right? PC, plastocyanin. When electron reaches to the plastocyanin, its energy goes down. Finally, that's why this arrow is downward, energy is going down. Finally, plastocyanin transfer electron to P700, which is called PS1. P700 again capture the energy from the rest of the antenna molecule and provide energy to the electron which is present in PS700. Electron again regain the energy from the light and it jumps. After jumping, this electron is received by its reaction center, its special chlorophyll molecule. Then it is transferred to the iron sulfur complex, iron sulfur protein, FES complex, right? From FES complex, it is transferred to the ferridoxin, FT, right? From ferridoxin, it is transferred finally, electron transferred to NADP plus. NADP plus, when it receive electron, it combined with hydrogen means reduces and form NADPH, right? And the formation of NADPH takes place. So try to understand. Final, that is the question for need. Who is the final electron acceptor? So NADP plus is the final electron acceptor. And this NADPH and ATP, which is produced in cytochrome B6F complex, these both are utilized in Calvin cycle. What is the Calvin cycle? Calvin cycle is a dark reaction. It is a process of CO2 fixation, right? And I have already explained you that 1 NADPH is equal to 3 ATP, energy equivalent. So this is 3 times more rich with energy. So this is non-cyclic. Why? Electron start its journey from one point and end up at one point, second another point. So this is non-cyclic. Electron do not come back. Tell me, and both are involved. PS1 and PS2 involved. Go through with it, and you have to try it because you will get the question from here. Cyclic, non-cyclic is the and what's the electric? These are the favorite question of teacher. First, tell me, is it clear, Alia, Siddhikan, Martin? Is that clear? Tell me.
Yes, sir, it's clear.
Okay. So, you have to note down only those points where I have undernote. Plastoquinine 1, plastoquinine second, pheophytin 1, plastoquinine PQ second, cytochrome B6F, right? Then plastocyanin, right? After that, this is the iron sulfur complex. So let's say 5 and ferridoxin 6. Rest you have to skip. Don't need not to note them, right? An electron transport chain, there you have nothing to like write down or nothing to dictate. Only the thing which you have to copy. Note down right now. You need not to make all the diagram.
Okay, sir.
You have to note down P680 reaction center, this H2O, 2H, half O2 electron, then this electron, then pheophytin, then plastoquinine, then cytochrome B6F, then plastocyanin. Then only you have to make P700, not this antenna complex, P700. Then plastoquinone, then iron sulfur complex, then ferridoxin and NAD. So that is the only thing which you have to draw. Okay, Martin?
Yes, sir.
Done? Or doing?
Done, sir.
Done. Very good. Martin? Done?
Yeah, done, sir. Yeah.
Okay, very good. Excellent. So here you have to draw this diagram. You have not to like write down anything, right? So this is non-cyclic because electron is not coming back, right? Try to understand my concept. What is the cyclic? Look, when Kelvin cycle is going on, then this will be non-cyclic. Means when NADP plus is present, then this will be non-cyclic. If NADP plus is not present, then what will happen? Who will receive the electron? There is no one to receive the electron. Suppose that one of your friend called you to like visit a particular place. I'll meet you over there, right? Or come to my home, right? I'll receive you without address. He or she called you. You went his or her home, and you are there. So you are non-cyclic because you have started journey from your home and you are staying with your friend. Non-cyclic. But suppose that he or she invited you, you started journey from your home, and then what happened? A moment when you reach the destination, he or she switched off the phone. What will happen? you will come back to your home. That is a cyclic, right? So that's how. So if NADP plus is present to receive the electron transport chain will be non-cyclic. When NADP plus is not present, then there is no one to receive the electron. Then electron will come back and cyclic will take place. That's why I am trying to this is the way I want to tell you, like what is the cyclic and what is non-cyclic mean and why it takes place. Did you got my concept? Why it takes place?
Yes, sir.
Come to the cyclic. When NADP, look, I have written here. When no NADP, what will happen? Electron will start its journey from P700. Electron have already reached to the P7. It will take the energy, it will jump. Primary electron acceptor, that is a special chlorophyll A molecule. So that is chlorophyll A molecule. It will receive the electron. It will transfer to ferredoxin. But from ferredoxin, NADP plus is not present. So from ferredoxin, it will again come back to plastocunine. From plastocunine, so when from ferredoxin to it will come plastocunine, formation of ATP will take place. Means energy will be generated, right? From plastocunine, it will rigid to cytochrome B6F complex, right? Right? Cytochrome B6F complex, right? Then again the ATP formation means energy generation will take place. Again, it will be taken by plastocyanin. From plastocyanin, it will come back to P700, right? So it have started its journey from P700, and again it have came back. Only the formation of energy taken place, right? So why this will take place when NADP plus is not present? So this is called cyclic photophosphorylation, right? Or cyclic electron transport system. Cyclic electron transport, right? So please note down. And it will take place will take place under low light. Let me know when you complete it.
Done, sir.
Please note down these 2 points as well. Right. First point, it will take place in low light intensity. This is the question of the NEET have been asked twice, right? When cyclic electron transport. Second, in absence of CO2 fixation, when light intensity will be low, CO2 fixation will not take place and NADPH is not required. When NADPH is not required, then only this will take place. So these are the 2 conditions when cyclic photophosphorylation will take place. Else, commonly, non-cyclic photophosphorylation will take place. Note down. Cyclic photophosphorylation will take place under 2 conditions. Done? Clear? Alia? Done?
Yes, sir.
So let's move to the next concept. What is a chemoosmotic? Try to understand. This is the best explanatory diagram. How does the what is chemoosmotic theory? Chemoosmotic theory or chemoosmotic hypothesis. This is basically a concept which explains how does the ATP formation take place when electron passes through with a particular substance or electron passes through particular electron carrier. How the ATP formation takes place in this reaction of photosynthesis, right? Electron transport. Try to understand. So when photolysis of water takes place, so this is a thylakoid, right? Try to understand. This is a diagram of a thylakoid. This is thylakoid membrane. Upper membrane, this is lower membrane. Around the thylakoid, there is a matrix, right? Matrix is present, right? This is called chloroplast. This matrix is called chloroplast. Right? What happens when water molecule break down, right? Then formation of hydrogen ion takes place, right? So due to formation of these hydrogen ion, the pH value decreases and hydrogen ion concentration increases inside the wood, where inside the this one thylakoid membrane. Then electron is transferred to photosystem setting. From photosystem setting, electron is transferred to plastocunine. From plastocunine, electron transfer to cytochrome B6F complex, right? When electron transfer to cytochrome B6F complex, on that time when electron is transferring, passing through the B6F, on that time 2 hydrogen ions are pumped in from outer matrix to inside the lumen of thylakoid membrane. 2 hydrogen are pumped. Due to the passage of these 2 electrons, 2 hydrogen ions are pumped in. Again, the hydrogen ion concentration increase. Then this electron transferred from B6F complex to plastocyanine. plastocyanin to again photosystem 1, then pyridoxine, then NADP, NADP plus will receive the electron and formation of NADPH will take place. Due to passage of these two breakdown of water molecule and due to passage of these electron from B6F, the hydrogen ion are pumped in. Due to the entry of extra hydrogen ion, what will happen? The hydrogen ion concentration will become high inside the lumen of thylakoid, and comparatively hydrogen ions are low outside of the thylakoid membrane. So between outer space and inner space, means between lumen and outer matrix, the hydrogen ion gradient will create. Why? What is the gradient? Outer side of the membrane, there is a low concentration of hydrogen ion, and inner side to the membrane, there is a high concentration of hydrogen ion. But unfortunately, hydrogen ion cannot pass through the membrane, means they cannot go outside. Why? Because this is impermeable for hydrogen ion. Normally, what happened? The movement of hydrogen ion should take place from its higher concentration to lower concentration. Means from the inner inside membrane to outer side membrane, from lower to from higher to lower concentration. But this is not taking place because this membrane is not permeable for hydrogen ion. So what will happen? There is only one space from where the hydrogen ion can leak out, right? This structure. This structure is called ATP synthase enzyme, right? ATP synthase enzyme is also present in the membrane of thylakoid. Now, due to the concentration gradient of hydrogen ion, what will happen? Hydrogen ion will start leaking out or passing out from lumen of membrane to the outer space or matrix. When hydrogen ion will be leak out, due to leakage of hydrogen ion, formation of ATP will take place from ADP. So in ATP synthase, the ATP will become ATP, right? And that's how the synthesis of ATP takes place. This so at the leakage of or at the leakage of every two hydrogen ion, one ATP will be produced, right? This is the mechanism of transfer or passage of electron. We call the chemoosmotic theory. Tell me, is it clear or not? This is very important. It will be full marker question, full size question. It will be if it will come at your school exam. And this is also one of the most favorite question of the teacher. Tell me, all of you. I want a response from all of you, Alia, Shidika, and Martin. Right? Did you got this concept or not? My first question. Tell me, Alia, Shidika, Martin, got it or not?
Yes, sir.
Alia, what's about you? And Martin, what's about you? Did you got this concept?
Yes.
Okay. So note down this one. So do not draw diagram like this way. Just make a membrane like this way. Look what you have to do. Make thylakoid, right? And this thylakoid you have drawn, then you can draw like this. Draw a structure, right? This is ES second, right? And then you have to draw that due to water molecule, H2O molecule, two electron and two hydrogen ion are passed out. Then like this, last two you need, right? Then cytochrome B6F complex cyto B6, sorry, B6F complex, right? Then plastocyanin, then photosystem 1, PS1, right? And then like this way, right? You have to draw like this way. So draw the diagram like this way quickly. And the shape of photosynthesis, this one, ATP synthase enzyme will be like this way. Same, like the way it look like, right? You have to draw quickly. Then I will dictate something for you because this question will come as a full 5-mark question in your exam, and it is important. What is important for 2-marker question? LHC1, LHC2, or photosystem 1, photosystem 2. Then what is the 3-marker question? Non-cyclic photophosphorescence. Then what is the 5-marker question? Chemosmotic theory. These 3 questions are very important for your exam, school exam. Yes, C3 cycle is also important. Done. Chidika? Okay, great. Martin, is it done? Martin?
Yes, sir.
Okay, great. So please note it down. Then uh, chemoosmotic hypothesis was proposed by chemoosmotic hypothesis was proposed by Michel in 1961. Michel in 1961. Electron transport both in respiration and photosynthesis. We will study the same concept in respiration in mitochondria, right? The same theory will be applicable for mitochondria as well. And the same theory is applicable for chloroplast, right? So electron transport in both respiration and photosynthesis produces proton gradient in bracket hydrogen ion. Proton gradient. Full stop. The gradient developed in outer and inner membrane space of halakoid lumen in chloroplast. Full stop. Next point. Lumen of thylakoid become enriched with hydrogen ion due to photolysis of water. Full stop. It also enriched by pumping in of hydrogen ion when electron passes through cytochrome B6F complex. Full stop. Next point. Because of these hydrogen sources, lumen of thylakoid develop high hydrogen ion high hydrogen ion concentration. Next point. Proton gradient is broken down. Proton gradient is broken down due to movement of proton through transmembrane channel transmembrane channel transmembrane channel, that is ATP synthase enzyme. Full stop. One molecule of ATP is formed. One molecule of ATP is formed at the passage of every two hydrogen ion through through ATPase enzyme. Clear? So this was the chemoosmotic theory, which is very important. Clear to everyone? Clear? Okay, great. Martin, is that clear to you? I didn't listen you. You switched on the mic, but I didn't got your voice. Anyway, get message. So look, whatever we studied till now, that was the part of light rays. This breakdown of the water is light rays. I have already told. That is the light. Now we have to study about the dark rays. Dark rays means fixation of carbon. How does the carbon dioxide fixation take place? Right one. The process of fixation of carbon dioxide was discovered by Kelvin Melvin, and that's why we call this cycle Kelvin. First thing, why it is called Kelvin. Kelvin cycle is also called C3 cycle. Why? Because the first stable compound is 3-carbon compound. This cycle, the first stable compound is 3-carbon. That's why it is also called C3 second one. Right? When Kelvin cycle complete its one round or one circle, then one carbon atom is fixed. Right? So, due to fixing of carbon dioxide, formation of glucose takes place. Right? So, glucose formula of glucose is C6H12O6. Formation of glucose, 6 carbon are required. And in one go of Kelvin cycle, only one carbon atom is fixed. So, Kelvin cycle will go on for the 6th time. Then formation of one glucose molecule takes place, right? So, when the 6th time Kelvin cycle goes on, then the formation of glucose takes place, right? So, this was the third. Now, come to the Kelvin cycle. What happened? There is an enzyme called ribulose biphosphate, right? Clear? Ribulose biphosphate, RUBP. It is a 5-carbon, right? RUBP capture or receive one carbon atom, one carbon dioxide molecule, right? This reaction, so this is the RUBP. You can see these purple color structure are carbon atom and these are the phosphate. Ribulose biphosphate. So there are 2 phosphate and 5 carbon, right? One carbon dioxide molecule will come. This reaction will be catalyzed by an enzyme called Rubisco. What is full form of Rubisco? Rubisco is known as RUBP carboxylase oxygen. This is called Rubisco. Rubisco, try to understand me. Rubisco is the most abundant protein present on the earth. And Rubisco is the most abundant enzyme present on the earth. Right. So this is the Rubisco. Right. So in the presence of RUBP, this reaction is catalyzed. Now what happened? 1 carbon this and 5 carbon this. Total becomes 6 carbon. Right. These 6 carbon molecule break down into 3 carbon molecule. 2 molecule of 3 carbon molecule. So at the reaction, formation of what happened? Formation of phosphoglycerate takes place. This is called 2 molecule of PGA. Phosphoglycerate. Phosphoglycerate is a first stable compound. It is 3 carbon compound. That's why it is called C3 cycle, right? And addition of carbon dioxide is called carboxylation, right? So carboxylation takes place. So RUBP is responsible for carboxylation. Then again, what happened? PGA is formed. So PGA, 2 molecule of PGA is formed. PGA is 3 carbon compound, right? Phosphoglycerate. Now what happened? 2 ATP break down into 2 ADP. We have studied the ATP formed in light reaction are utilized in dark reaction, right? Calvin cycle. I have already explained you, right? And even NADPH produced in light reaction are utilized over here. So what happened? 2 ATP will break down into 2 ADP, right? Then 2 NADPH will break down into 2 NADP plus and energy is generated. Then due to this reduction will take place and from phosphoglycerate formation of 2 molecule of glyceraldehyde phosphate take place. In short, this is called G3P. So 2 molecule of G3P is formed, right? Again, they are also the 3 carbon compound. What happened from here? So 2 3s are 6. So total 6 carbon compound. Again, what happened? 1 carbon out of these 6 carbon, 1 carbon is transferred to the for the formation of glucose, right? And now out of these 6, 1 carbon is transferred to the glucose. Then only 5 carbon are left behind, right? These 5 carbon will make ribulose phosphate RP, right? This is called regeneration. This reaction is called regeneration because again formation of ribulose phosphate takes place. Then again, 1 ATP will break down into ADP and it will give phosphate molecule or phosphate atom. From ribulose phosphate, formation of ribulose biphosphate will take place. So this complete reaction is called Calvin cycle. Due to Calvin cycle, this 1 carbon dioxide molecule have given 1 carbon atom which have been transferred or fixed in the form of glucose. So when 6 times this Calvin cycle will go on, 6 carbon atom will be fixed and formation of 1 glucose molecule take place. So in the nutshell, for the formation of 1 glucose molecule, at least this cycle have to go on 6 times. Tell me if is there any ambiguity or if is there any doubt if you have any doubt, ask. Edukiran Martin, tell me, Martin, is it clear or you have any
Yes,
doubt?
sir, it's clear.
Okay, Martin, what's about you?
Yeah, clear.
Okay, so please note down right now. The only thing is, you need not to note down these structure, purple color and yellow color molecules, right? This complete cycle you have to know. because you have to draw this cycle only. If you get the question, draw the well-level diagram of or schematic diagram of Kelvin cycle, then you have to draw this cycle only. Not that one which is given in your book. I have a question. Which is the primary acceptor of carbon dioxide? Which is the first acceptor of carbon dioxide? Sidika? In Kelvin cycle? Yes, Shidika. Tell me. Who is receiving carbon dioxide over here? No, not PJ. PJ is first compound. Carbon dioxide received by RUBP. So RUBP is the primary acceptor of carbon dioxide. That is the question of need. Right? RUBP is the first acceptor. PJ is the first stable compound of C3 cycle. Note down right now.
Done, sir.
Martin, that done?
Yeah.
So write out. Kelvin cycle. Kelvin cycle can be Kelvin cycle can be explained in 3 steps. First, carboxylation. Second, reduction. And third, regeneration. Full stop. First subheading, carboxylation. This I will explain this onward because in this case I have left. So Martin, next class we'll start from here. Right? I will explain you what is the carboxylation, reduction, and regeneration. Right? I'll dictate.
Yeah.
Okay. So see you in next class.
Thank you.
Yeah. We'll try and revise it, right? And we'll see you next class.